So, in the previous discussion, we looked at 4 major features of mutation, and in this discussion, we will cover the remaining 3. So, the first feature that we are going to talk about, a very well known feature about mutation, is that they are random with respect to what the organism needs. Now, what do I mean by that? Now, suppose you have a petri plate, and in that petri plate, you have put some culture of, say, some bacteria, which is let us assume sensitive to some antibiotic or a bacteriophage or whatever. Bacteriophages are viruses that kill bacteria. So, when you do that and when you spread it and when you incubate it for say 24 hours or 48 hours, you are going to get something like this, where you will get lots of colonies of the bacteria on the plate. Now, suppose you hit this with some antibiotic or some bacteriophage, and what will happen? Most of the bacteria will die, most of the colonies, but you might end up getting something like this, where a few colonies will survive. Now, obviously, these colonies have some kind of a mutation which allows them to survive the challenge. Now, here comes the question. What exactly is the mechanism or way in which these mutations have arisen? There are 2 ways in which this can happen. One is that the mutations arose after they were challenged with the bacteria in response to the challenged with the antibiotic or the phage. So, the mutations are a direct result of that challenge of that you have subjected them to. And the other possibility is that the mutations were already there in the bacterial cells before they were plated. Obviously, they are not there in all of them, they were there in some of the colonies, and when you hit them with this challenge, then only those survived which had the mutation and all the others died out. Now, how do you distinguish between these 2 possibilities? Now, although this looks like a very simple question, actually this is a very big and well known question in biology, and the primary reason for that is if the mutations are arising after the bacteria are being challenged or the organism are being challenged with some environmental factor, then one can say that adaptation, which is a major feature of most organisms, that can be directly a result of the mutation. You do not really need natural selection to be the causal reason for adaptation. So, if the first hypothesis is correct, then mutations will become a very major evolutionary force which leads to adaptation. But if that one is not correct, then of course what Darwin said about natural selection will end up getting a lot more credits. So, that is why this was a very big question in biology, and how the distinction between these 2 hypotheses was made for the first time by these 2 gentlemen, Max Delbruck and Salvador Y Luria. And what they came up with was this thing called the fluctuation test, a very elegant experiment, and which ultimately led to a Nobel Prize in 1969. So, you can see the importance of figuring out the answer to that particular question. Now, the fluctuation test is a very important technique, and it is still used in certain areas to estimate the mutation rates. However, it is a slightly complicated technique based on properties of statistical distributions. Therefore, in the context of this course, we are not going to look at this particular experiment. We will look at another experiment which is equally famous, also leads to a Nobel Prize, but which is much more simpler to explain and is very elegant, logically elegant. And that is the experiment performed by the husband and wife team of Joshua and Esther Lederberg, and I am sure all of you would have heard about this. This is the famous replica plating experiment. So, what did they do? So, they started with populations of E. coli that were sensitive to a certain bacteriophage known as a T1 phage. So, they took such a clonal population and they let it grow, and then at some point from the liquid culture they plated it on a petri plate and obtained a bunch of colonies exactly like this. So, this is what the original plate on which they plated the E. coli, they called it the master plate. And of course, on this master plate the colonies have a certain spatial distribution. Now, they did a very elegant innovation I am not even sure if the innovation is theirs, but certainly they used it in this particular context, and the innovation was very simple. This, all they did was they took a plastic cylinder and then they took a cloth, a velveteen cloth, basically some kind of a rough cloth, and they sterilized the cloth, they sterilized the cylinder, and then they put the cloth on top of the cylinder with some kind of a tying a rubber band or some kind of a thread. So the main thing is that both the cloth and the cylinder, both of them are completely sterile. So what they did was they took this thing apparatus and they took their master plate and they simply pressed that velvet cloth on the master plate. What will happen? You have all these colonies, the cloth is rough. So from each colony some number of cells are going to go and stick to the cloth, right? Now they took this cloth with the bacteria and they took fresh Petri plates containing bacterial medium and they pressed them carefully on those plates. What will happen? From each point where there were some bacteria are going to get transferred to this cloth, to this new Petri plate. Now the most important part over here is, if you are doing it very carefully, then the pattern of the colonies that you find over here in the on the master plate, that pattern of the colony is going to get replicated in the fresh plate to which you are pressing the velveteen cylinder. In other words, these the positions of these colonies will become the same and you can do it multiple times. So these are the so called replica plates, plates in which you have exactly the same colonies replicated onto the on a different fresh plate. Now once you have the replica plates, you can now ask the question that we asked, how to distinguish between the two hypotheses. What was our first hypothesis? Our first hypothesis was that the mutations arose after the bacteria were exposed to the phage. So what they did, they had the master plate, they created the replica plates and then they added the virus. Now remember, the mutations arising is a probabilistic event, right? So not every bacterial colony which is getting challenged is going to lead to a mutation. Therefore, if it is probabilistic thing, then you do not really expect that the same colonies again and again are going to get the mutations. In other words, the mutations are going to be formed in some colonies, but these are going to be different colonies for different replica plates, which means that the positions of the colonies are going to be very, very different. In other words, if you have the mutations arising after exposure to the virus, the resistant colonies will be at different positions on the plate. Now what would have happened if the mutations arose prior to the exposure to the phage? If they have arisen prior to the exposure to the phage, then they are already there in the master plate, right? Now if they are already there in the master plate, then of course they are also already there on our replica plates. And more importantly, because the replica plate is spatially a copy of the master plate, therefore they are exactly in the same position. And hence when you add the virus over here, then what is going to happen? All the resistant colonies are going to be found at the same position on all the replica plates, right? And that when Lederberg, when the Lederbergs did the experiment, that is exactly what they ended up finding, which told us that the second hypothesis is correct. All the mutations were already, the resistant mutations were already there on the master plate even before they had been exposed to the phage. So the implication of this is that the mutations arise irrespective of what the organism needs. Remember, at the point at which the mutations arose on the master plate, at that point the bacteria had not met the phages. So there was no requirement for the phage, they arose independent of that anyway. And in that context, the mutations are random to fitness, random to the benefit or harm of the organism. Now, as I said, if this were not true, one could have explained adaptation through mutation. In other words, one could have said organisms are adapted to their environment because each time the environment throws a challenge, the organism comes up with a new mutation. Now, of course, there is a very simple logical problem with this. How on earth will the organism know which mutation to come up with, right? So there are so many genes, there is no way for the organism to figure out, okay, now I am being challenged with let us say rifampicin, therefore I need to mutate the rifampicin gene in my body. Doesn't really happen. I mean, one cannot conceive of a way in which it can happen, but even if it could have happened, the replica plating experiment is showing that actually it doesn't. Okay. So adaptation cannot be explained through mutation, and hence Darwin's theory of adaptation through natural selection got a lot more credence. However, the big thing to remember is, although mutations are random with respect to fitness, that doesn't really mean that they are unbiased in other ways. Actually, one can see that there are many ways in which mutations can be and are actually biased. I will give you just 3 examples. So, for example, remember we talked about transition mutations and transversion mutations. So, transition mutations are purine to purine, pyrimidine to pyrimidine, whereas transversion mutations are purine to pyrimidine and pyrimidine to purine. So, if you remember that square figure that we had, you will immediately realize that there are more ways to have transversion mutations than transition mutations. Yet, if you look at the rates, transition mutations are the ones which are actually much more common than the transversion mutations. Similarly, if you look at our genome, it is not that the genome has equal probability of mutation at all the positions, absolutely not. There are certain locations on the genome which have much greater mutation rates than other locations, and these locations are known as mutational hotspots. So, the presence of these hotspots straight away tell you that there is some degree of spatial bias over the genome in terms of mutation rate. And finally, in many species, male germline mutation rates are actually much greater than female germline mutation rates. So, as I said, these are just 3 examples of mutational biases. There are many other kinds of biases that people have uncovered. The other major feature of mutation is that mutation is a weak evolutionary force. Now, what do I mean by that? Now, in order to understand the meaning of this, it will be better if we go through an example. So, let us assume that you have a population, we are looking at one particular locus, and let us assume that to begin with the population only has the allele A2, and at some point the new allele A1 has freshly arisen in the population. And let us assume that right at the beginning the frequency of this A1 allele is 0.001. Now, you can very logically ask that if in a largish population you have just one mutation freshly arising, how is it that the mutation frequency will be 0.001? Shouldn't it be much smaller than that? And you are absolutely right. In any slightly largish population, if you have a fresh mutation, it will be much smaller, the frequency will be much smaller. However, we are just making a case, okay, it is a theoretical example that we are taking. You will realize later that whatever case I am making becomes much stronger if we take mutation rates much lower than 0.001. Now, the question that we are asking is that suppose from 0.001 this mutation needs to reach a frequency of 0.1, okay, which is not a very high frequency, but let us say it has to go to 0.1. How long will this mutation take to reach that frequency if the rate of forward and reverse mutations are 5 into 10 to the power minus 5. Now, what is forward mutation? Forward mutation is A1 converting to A2. What is reverse mutation? Reverse mutation reverse mutation is A1 becoming A2. Okay. So, I have ended up writing the same thing over here. So, forward mutation is A1 becoming A2 and the reverse mutation is A2 becoming A1. So, this should be 1 and this should be 2. Yeah, A1 becoming A2 and this is A1. Sorry, this is a typo over here. So, anyway, now why am I choosing a rate of 5 into 10 to the power minus 5? So, turns out that the rates of mutations roughly vary from 10 to the power minus 3 in RNA viruses, which are the most highly mutating organisms, to about 10 to the power minus 10 or 10 to the power minus 11 or so. That is the whole range. So, I have simply taken something in the middle of that, about 10 to the power minus 5, right. And let us assume that all the other conditions of Hardy-Weinberg equilibrium are operative. Now, how will one even solve this kind of an issue, kind of a problem? So, in order to do this, what we will do is we will do a very simple simulation. So, this is a link which contains a very nicely done online simulation platform. This is from the same biointeractive platform that we met earlier. So, what we will do is we will go there. So, it is free to everybody, so you can play with it later and have fun with it. So, let us go here. Okay. So, this is the platform, and when you come here, this is where you should go, Population Genetics Explorer Start Tool. When you do that, it will open a new tab, and on this tab it has all kinds of information, but you can check those out later. So, it gives you two kinds of simulations, one where you just do individual simulations, one case at a time, and the other where you do replicated simulation, you run multiple replicates at the same time. So, right now we are going to do individual simulations. Okay. So, here is the setup for the simulation. So, first thing it asks is population size. So, remember we said that all other conditions of Hardy-Weinberg are operative, and therefore, we will set the population size at infinity, right. So, we will click it over here. How many generations? We can keep it at 500, does not really matter. We will change it later in a few minutes. And starting frequency we said is 0.001, and now I will tell you why we took 0.001, because that is the least count of this particular simulator. Okay. If you are doing your own simulation using Excel or Python or something, you can obviously keep it at a much lower value. So, here the lowest value is 0.001. Great. Now, we need to do some additional settings. What is it that we are studying? We are studying mutation. So, we click on mutation. So, here it gives me the forward mutation rate and the reverse mutation rate. So, I have to do this 5 and I will put this at 5. This is already at 10 to the power minus 5, but we can alter them to whatever we want, right. So, in the context of our question, we have put them at 5 into 10 to the power minus 5, and then all you need to do is to say run simulation. Remember, our question is how long does it take to go from 0.001 to 0.1. Run simulation. 500 generations are done, and if you just take your cursor over here, it tells you where the values are, and okay, so you can see it's even after 500 generations it's 0.025, so it has not really reached 0.1. Okay, so what will we do? We will go up and we will change the number of generations. So, let us make it the highest that the platform can take, which is 10,000. So, we do that, we come down and we say run simulation again. As you can see, it is running pretty fast, and here now you can see some movement, and let us see where does it hit 0.1. So, if you look at it, it hits 0.1 somewhere around 2329 generations, right. Now, 2329 generations is actually a very, very big number. So, what this is telling you is that assuming that there is no selective advantage for the mutation, it does not give any benefit or it does not cause any harm or anything, even then with those mutation rates, this is going to take this many number of generations, which is a very large number. So, this is what I meant when I said that it is a weak evolutionary force. In other words, it will cause a change, but it will cause the change very, very slow. So, as I said, 5 into 10 to the power minus 5 is still a slightly highish number. Okay. What happens if I reduce the mutation rate to, let us say, 10 to the power minus 7 or 10 to the power minus 9. So, let us do 10 to the power minus 7 first. We are still keeping the forward and the backward rates similar. So, if you have reduced the mutation rates, what do you think? Should it take more time or should it take less time to reach 0.1? Quickly make a guess and let us run and see it. And you can see that even after 10,000 generations, this has only gone up to 0.006. So, obviously, this is taking a lot more time. Yeah, because we are starting from a very small frequency, we are trying to go high. If you are reducing the mutation rate, it will take longer. So, by that token, if we increase the mutation rate, it should take smaller amount of time. So, let us say, let us make it 10 to the power minus 4 equal for both, and we run the simulation, and there you go. So, you can see that it reaches this 0.1 much faster, it reaches it about 10 times faster in about 234 generations or so, right. So, you can see that when the mutation rates are similar, it is taking some amount of time, quite some time, and the smaller the mutation rate is, it is taking larger and larger time. And if we are in biologically relevant zones, which is like 10 to the power 6, minus 6, minus 7 and so, it is going to take very, very long time. Now, here lots of students ask the question that if I have the same backward and the forward mutation rate, then why is it that the mutation rate is the allele frequency is changing at all? Because shouldn't it be the case that the allele frequency, if the forward and mutation rates are the same, then the allele frequency should not change to begin with? That is not really true, because remember, you have, when the frequency of the A1 allele is 0.001, then the frequency of the other one is 0.999. So, at that point, even if the frequency mutation rates are the same, the actual number of alleles which are turning from A1 to A2 are very small, because the forward mutation rate, but the number of alleles which are turning from A2 to A1, that number is going to be very high, because even if the mutation rates are the same, it is being multiplied by a larger number, okay, larger frequency. And that is why during the initial stage, the rate at which it is the rare allele is going to pick up is going to be pretty fast, and then the closer their frequencies come to each other. So, basically the closer P and Q are to each other, the number of alleles that go backward and forward, assuming a same forward backward mutation rate, that becomes similar. Okay. So, I will show that to you in a minute what I mean. However, there is something else which is very interesting over here. What is that? You can see that in this particular case, this is stabilizing at some point, and if you just take it forward, you will realize that it is stabilizing somewhere close to 0.5. Okay. In fact, it stabilizes exactly at 0.5, as I will show you by slightly increasing the mutation rate. So, let us say, let us make it 10 to the power minus 3. Okay. Both forward reverse are the same, and when we do that, you can see that it stabilizes, and it stabilizes at 0.5. Now, let us make it something else. Let us go back to 10 to the power minus 4, let us make it say 9 into 10 to the power minus 4. Okay. And we run the simulation again, you can again see that it stabilizes at 0.5. So, this property, although I am showing it to you for 10 to the power minus 4, this property is actually true for any time the forward and the backward mutation, the reverse mutation rates are the same. Now, why is that? In order to understand why that is so, we need to go back to our PowerPoint and see how exactly the mutation rates behave at equilibrium, or rather the allele frequencies behave at equilibrium. So, let us assume that our two alleles are A1 and A2. The rate at which A1 is becoming A2 is U, and the rate at which A2 is becoming A1 is, let us say, V. Okay. So, let us further assume that the initial frequency of this is given by P naught, and the initial frequency of this is given by 1 minus P naught. You can also say 1 minus P naught equal to Q naught. Okay. Now, we want to look at delta P. What is the rate at which the value of P, the frequency of the A1 allele, is changing? Remember, that is what we are interested in. Now, this thing is equal to the rate at which A1 allele is being created minus the rate at which A1 allele is being destroyed. Now, what is the rate at which A1 allele is being created? Remember, A2 is becoming A1, at a rate v, right. So, A1 is being created at a rate v into 1 minus p naught, correct, and A1 is becoming A2, that is A1 is becoming not A1 at a rate which is per capital rate is u and multiplied by p, right. So, p naught at this point. So, this is the expression. Now, what happens at equilibrium? At equilibrium, delta p has to be equal to 0. So, at equilibrium, delta p is equal to 0, which implies that v minus, so now we will not call it p0 anymore because this is at equilibrium, so we will call it just p. So, v minus vp minus up is equal to 0, right. So, what do I need to do? I will end up taking p as common, rather minus p as common, v minus p. So, this becomes v plus u, okay, is equal to 0, and I can take the p to the other side. So, p into v plus u is equal to v, which implies that p at equilibrium is equal, so I am just calling it a p hat, p at equilibrium is equal to v by v plus u, okay. This is the relationship, this is the general relationship. Now, the special case that we were doing was v equal to u, right. So, when you have v equal to u, then p hat is equal to v by v plus v equal to v by 2v. So, you just cancel these two out, right. This is why we were obtaining that thing, that when the forward and the reverse mutation rates were the same, then the whole population was stabilizing at equal frequency of the two alleles. Now, what happens if the allele frequencies are not equal? That you can figure out from this particular relationship. So, what are the implications of this? Remember, biologically speaking, the range in which things go is minus 10 to the power minus 3 to 10 to the power minus 10, 11 or so, and 10 to the power minus 3 is only for the exceptionally fast RNA viruses and so on. So, mostly things are in the range of 10 to the power minus 7, minus 10 to the power minus 5, minus 7, minus 9, that zone. In that zone, by itself mutation will change the allele frequencies very, very slowly. However, when mutation operates in conjunction with other evolutionary forces, particularly selection, mutation becomes a very, very potent force, and we are going to look at that as we go along. However, before we get into that, we first have to study selection, but before we study selection, we have to look at one other thing which is very important. So, till now we were mainly talking about mutations in the genes, etc. But I also told you that one of the things about EES is that we have now started realizing that there are lots of inheritances that are happening which are non-genetic, not in our ATGC sequence. So, before we go into other evolutionary forces, we need to take a quick digression and look at what these other non-genetic inheritances are and in what ways they can be effective in terms of shaping our evolutionary trajectories. So, that is something that we are going to do in our next discussion. See you.